Here I'll cover what is chemical oxygen demand, the characteristic of COD parameter, and what is this chemical oxygen demand. Say the sewage or the waste content which present in the water, normally considered sewage. Sewage is a liquid waste which includes human and household wastewater. There are two parameters used to find the waste or the load of waste which present in the water. They are a biological oxygen demand and the chemical oxygen demand. The waste may be a domestic sewage or industrial sewage. This waste usually degrades or they get oxidized in the presence of dissolved oxygen or oxygen is present in the water. So to find the organic, inorganic or the waste load in the water, we normally use the two parameters called biological oxygen demand, in short BOD, and the other one is a chemical oxygen demand. The biological oxygen demand may defined, it is the number of milligram of dissolved oxygen. Here we consider dissolved oxygen, the oxygen which is present in the water, required for biological oxidation of oxidizable impurities present in 1 liter or 1000 ml of water sample under, this is the condition, aerobic condition at the 25 degrees Celsius for a period of 5 days. That is the amount of oxygen for the biological oxidation of all the oxidizable impurities present, okay, in 1 liter of or 1000 ml of water under aerobic condition at 25 degrees Celsius for a period of 5 days. And usually the BOD is expressed in terms of ppm of oxygen. It is expressed in terms of ppm of oxygen. And this is not empirical. Sorry, this is empirical. Okay, the main thing is it is empirical and semi-quantitative. It is not accurate. It is a semi-quantitative method. It represents only biodegradable impurities under aerobic condition. Determination is a slow and time consuming. To improve this, it is only the amount of decomposable organic matters which present in the sewage you can detect by biological oxygen demand. And the determination mainly deals with the determining the dissolved oxygen content after and before incubation period of 5 days, so that it takes a lot of time. So the other method used to predict the organic load which is in the water called it as a chemical oxygen demand, COD. So BOD value takes into account only biologically degradable or oxidizable impurities and does not account for biologically non-oxidizable or very slow oxidizable impurities. To overcome these difficulties, we'll use another parameter called chemical oxygen demand. It represents total oxidizable impurities present in a sewage water. This includes both biologically oxidizable as well as biologically inert and chemically oxidizable impurities or organic matter which present in the sewage water. So we defined chemical oxygen demand is the amount of oxygen equivalent of acidified potassium dichromate required for the chemical oxidation of total organic load present in 1 liter of water sample. It is the amount of oxygen equivalent of acidified potassium dichromate. This chemical is used to produce oxygen acidified potassium dichromate required for the chemical oxidation of all the impurities present in 1 liter of wastewater. So this method, the COD method, is a satisfactory quantitative method and it measures the total organic load. And it is preferable to BOD as the results are reliable, a rapidly measurable parameter. Within 3 hours, we can predict the total organic load which present in the water. Then always COD is greater than BOD because it tells the total organic load. Okay, and determination of COD is a very simple method. It's an indirect method. It is not possible when you treat the potassium dichromate with the water. See, the potassium dichromate releases oxygen and that oxygen is utilized by the waste which is present in the water sample and forming a stable product. How? When you introduce a potassium dichromate, particularly acidified potassium dichromate, it liberates nascent oxygen. And the organic waste normally contains carbon, hydrogen, sulfur, nitrogen like component interact with the oxygen forming a stable compounds. Okay, it forms a stable compounds. Okay, like nitrate, nitro, sulfur dioxide, etc. It forms. This is a aerobic condition. See, the quantity of oxygen utilized for the oxidation of these impurities is exactly equal to the COD of wastewater. Once the oxygen is trapped by these molecules, it will not available for further reaction, this oxygen. So that's why we want to carry one blank titration. We'll take a distilled water in place of wastewater. This is a wastewater usually contain these component. Okay, wastewater contains this component. So if you can take a distilled water, so it will not contain any component and is treated with the same potassium dichromate, and this net reflects the total quantity of K2Cr2O present. And this minus the utilized potassium dichromate, so leads to the gives the actual potassium dichromate consumed by the organic waste. So here, that's why the sample water is reflexed with the known excess of potassium dichromate. We'll take a sample water in a RB flask. Okay, we'll take a sample water in a RB flask. Okay, and it is reflexed using a condenser. That is a reflexing method. Is we follow the reflexing method in an acidic medium in the presence of silver sulfate and mercuric sulfate. The silver sulfate act as a catalyst. It act as a catalyst and mercuric sulfate avoid the interference of chloride ions. Otherwise, the chloride ion combined with the added silver sulfate and prevents the activity. Silver chloride is not a catalyst. Only silver sulfate acts as a catalyst. So silver sulfate helps for the easy oxidation of straight run organic compounds, pyridine, and even aromatic compounds so that the mercuric sulfide avoid the interference of chloride ion. As a result, the potassium dichromate oxidizes all the oxidizable impurities to respective compound. Okay. Then the amount of here it consumes some amount of potassium dichromate. Say you are using some highest excess quantity of potassium dichromate. In this, a portion of potassium dichromate is utilized by these organic component. Okay, then there is some component is unconsumed. Some of the amount of potassium dichromate is unconsumed. So we will detect the unconsumed potassium potassium dichromate by titrating it with the ferrous ammonium sulfate. This is already consumed for the oxidation of this organic compound, but this unconsumed potassium dichromate is determined by titrating with FAS using ferroin as an indicator. So ferroin, the chemical name is 1,10-phenanthroline iron 2 complex. And it has the capacity to show the color change when it is in contact with the oxidizing environment, it impart bluish green color, and when it interact with the reducing environment, it impart red color. So that's why the 110 phenanthroline ion to the complex ion will take 2. When it is in a 2 state, it impart red in color. When it is in a 3 state, that is when in the oxidizing environment, so it imparts a blue color or bluish green color. In a reducing atmosphere, it impart red color. Okay, so when the color changes to red, that indicates all potassium dichromate is reacts with the reduced by a ferrous ammonium sulfate, so that the consumed potassium dichromate, then we calculate. First we detect the unconsumed potassium dichromate in terms of FAS. Okay, so once you know the consumed potassium dichromate, we can able to find the unconsumed potassium dichromate by treating with the distilled water. Same way, the consumed potassium dichromate can be calculated, which corresponds to the COD of the sewage water. And what kind of reaction take place? Potassium dichromate in acidic medium forming potassium sulfate, chromic sulfate, water, and 3 moles of nascent oxygen. And this nascent oxygen is utilized by the organic waste which present in the sewage water. Organic waste which present in the sewage water. But still, the some amount of nascent oxygen becomes unreacted. A reacted is utilized by the organic, means unreacted is remains as it is. And this unreacted potassium dichromate titrated with the ferrous ammonium sulfate forming ferric sulfate. It consumed oxygen. The quantity of oxygen consumed is exactly equal to the unreacted oxygen which present in the sample. Okay. Potassium dichromate react with the ferrous sulfate in the presence of sulfuric acid forming potassium sulfate, chromic sulfate, ferric sulfate and water. The procedure what we follow is, see here we normally detect the consumed potassium dichromate, unconsumed potassium dichromate first. Then to predict consumed potassium dichromate, we have to carry the titration with the distilled water. Same, the potassium dichromate is treated same way we will treat with the distilled water, and that distilled water is titrated against FAS. We will get the net quantity of FAS used for the oxidation process. So that net quantity minus the unconsumed potassium dichromate gives the consumed potassium dichromate. See, first thing we have to prepare a standard ferrous ammonium sulfate. For that purpose, we have to use ferrous ammonium sulfate crystal that is transferred into 250 ml standard flask. Usually, we'll take 2.5 gram of ferrous ammonium sulfate to prepare 250 ml solution. To that, before adding water, we have to introduce 1 test tube full of sulfuric acid to prevent hydrolysis. Otherwise, this ferrous sulfate combined with the sulfuric acid, okay, forming the ferrous sulfate. If you are not introduced sulfuric acid, the ferrous sulfate consumed with the water forming ferrous hydroxide. Okay, the ferrous hydroxide is not act as a reducing agent. So we have to prevent this reaction by adding sulfuric acid. That's why 1 full test tube of dilute sulfuric acid is added. Then small quantity of distilled water is added. Once the crystal is completely dissolved, the solution is made up to the mark with the distilled water and shaken. Then you have to carry a back titration. For the determination of back titration, we have to take a 250 ml of water sample, and to that we have to introduce 1 gram of silver sulfate and 1 gram of mercuric sulfate in an RB flask. Then that mixture is refluxed for about 2 to 3 hours, around 2 hours, refluxed for about 2 hours. Then cool to room temperature. Then from that, say 25 ml, known volume of this waste sample is titrated against standard FAS using ferroin as an indicator. Before that, we have to introduce around half test tube of concentrated sulfuric acid just to make the medium acidic. Okay, say take 25 ml of that refluxed solution, water, refluxed water, then half test tube of concentrated sulfuric acid, 2 to 3 drops of ferroin indicator that contain titrated against FAS taken in the burette till the color changes from blue green to reddish brown. And you have to note down the volume of FAS consumed by unconsumed potassium dichromate. So once you detect the unconsumed potassium dichromate, you have to carry a blank titration. Say for blank titration, the procedure is repeated by taking the same volume of distilled water in place of sample water. Then calculation, you have a tabulation column and thereby you can detect the volume of FAS consumed by sample titration. That is volume of standard FAS used by an unconsumed potassium dichromate. It is unconsumed K2Cr2O7. That is AML. Volume of water sample taken is B1 ml. Volume of FAS consumed by unconsumed sample is AML. Then volume of FAS consumed by a blank titration is BML. And this amount of potassium dichromate consumed in satisfying COD in terms of FAS becomes B minus AML. And you know the normality of FAS is N, so that we know the standard equation N1 V1, and this one is considered for water, and this one is for FAS. Okay, so N1 normality of COD of the sample N1 is equal to N normality of FAS into B minus blank titer value minus sample titer value divided by V1 gives the normality of the water sample. Once you know the normality of the water sample, you can able to calculate the COD in terms of ppm, oxygen equivalent. So COD of the sample becomes normality into B minus A N 8, that is to convert into equivalent or like ppm, that is expressed in terms of equivalent weight of oxygen. Okay, that's why we'll take multiply by 8, and 1000 is to convert into in terms of liter, gram, milligram. Okay, milligram per gram per dmq, or a milligram per, you have to convert into milligram per dmq. So gram to milligram conversion is multiplied by 1000. So this gives the COD of the wastewater. Okay, in this way we can able to find the COD of the sample. Thank you.